Also in the Davis lab, and he's going to talk to us about some approaches for normalization and for dealing with variability, especially like longitudinal batch variability in data, in large data sets. So I'll just mention now, because when we're all done, we're going to go to lunch, that what we will do in the afternoon is we will meet in CCSR in the basement. There are 3 rooms that are listed on your schedule for the 3 different demos. And I'm going to allow you to just self-select, okay? So you can go to any one of those 3 rooms to start off with. But I'd like to sort of have no more than 8 people per group so that the groups are evenly divided. So if you come after lunch and you go into one of the rooms and there's less than 8 people, that's where you can be. If there's already 8 or more, then go to another room and we'll just select ourselves. After 1 hour, when one set of demos is done, everybody will rotate to the next and then the next. So after 3 rotations or 2 rotations, you will have seen all 3 demos. That's how it's going to work. Okay, Brian.
Thanks. Can everyone hear me okay? Or do I speak back there? Okay, so I'm going to talk about the quality control and sort of management up front on large data sets. And since I'm the person standing between you and lunch, I'll make this brief, but it'll be hopefully informative. So just as a quick outline, it's going to be short, but in terms of the topics covered, as you'll see, it's kind of this game where you're doing the same thing over and over again. It just depends on what the data is, and you should have some familiarity with that data. But as you all know, there's been some big changes in terms of how data goes through the pipeline from collection to ultimately analysis and interpretation. And as we've seen, the data pipeline here at the HIMC is fairly sophisticated, handles lots of high-throughput data. But just to contrast what used to happen in the old days, in the bad old days, you would typically collect maybe in 1 tube or multiple tubes, and then you'd put it into some black box that would run the assay. And typically it was a single output, right? So you get one readout on whatever you're interested in, and then you, depending on, I don't know how sophisticated you guys are or not, you might run a simple t-test or something like that to discriminate between the differences in the readout. And then you would just plot that difference maybe on a scatter plot, and so it'd be fairly simple, you know, change in expression over time or differences between 2 samples and so forth. And if you look at kind of the effort required in this, most of the effort was on the sort of front end of things, right? The collection is hard. Sometimes the assay took a long time, but the analysis and interpretation was fairly straightforward. When you're only dealing with a single readout, it's not so difficult to interpret the results. You can set up the experiment in ways that it's easy to design the experiments. Nowadays, though, however, we typically deal in large formats, whether they're those 96-well plates or multiples thereof 96-well plates, as you'll see. And the assays have become more sophisticated. And you guys have already talked about and seen presentations on the sophisticated assays like CyTOF or PhosphoFlow. And so they're putting out multiple readouts. And this was true for gene expression. And so now the question is, how do you actually analyze this data? I mean, this is now multiple, thousands and thousands of readouts potentially. So it's not such a straightforward, simple t-test one against the other. And even more complicated has become sort of the visualization and interpretation. And I don't know if any of you have seen these kind of hairball correlation plots, but it's very easy to generate this nowadays, but it doesn't really give you much information. It's hard to interpret. And so really, if you, not to necessarily downplay the collection and assay efforts, but this effort has really gone down in terms of the data lifetime, or the lifetime of your data. And the effort is really now on analysis and interpretation, okay? And so we've really moved, and this is how I think of, and others as well, we're moving in science from a place where it was really hard to get data, and the questions were really driven by what data you could collect, to now it's easy to get data, and it's really, we're hypothesis limited, or analysis limited. And I've heard people from Gary Nolan's lab talk about this analysis fatigue, and that's partially why Cytobank was created, to try to sort of minimize the upfront cost of all the analysis that has to go into this now multidimensional and high throughput data. Okay? And we've had gene chips have been around for a long time. These are Illumina gene chips, so these typically generate on the order of 40 to 50,000 probes, depending on what you're doing. Luminex, you can measure 50 cytokines at a single time. With phosphoflow, you can readily measure, you'll see, hundreds of different combinations. And with CyTOF, it's on the same order in terms of cell types and different functionalities. So it's really, these high throughput methodologies have pushed now back on the computational biologist and bioinformaticist like myself to sort of how do you actually facilitate the analysis. Okay? And so what I'm going to talk about today is kind of the simple distillations for the upfront of analysis for you guys that will hopefully make things easier to sort of get started on interpreting your data, make sense of what has come off the machine, and then go forward with more sophisticated analysis and visualization. Okay? So as I've said, immunological data sets now are quite rich. They're probably some of the richest in terms of biological information. And so we're routinely measuring on the order of tens of thousands or millions of different data points, depending on what either tissue or molecules you're looking at. And so with all of these, which you've talked about over the last couple of days, it has become kind of a big problem in what do you do with all this information and how do you handle it. And even more to the point is we now have the ability to collect many samples and then run those samples in a high throughput manner. So it's not like you're doing a few, but you can do hundreds of samples now routinely. And so it creates this situation where you have to often break up samples into batches, and that then creates some problems with, you have batch problems as well as the fact that you're collecting just so much data. Okay? So in terms of the general scheme of things in this kind of new data pipeline, I'm going to talk about the smallest slice, and this is coming basically right after whatever's come off the machine. So there's a lot more we could talk about in terms of what types of analyses to do. Those are typically question specific in terms of what biological questions you're interested in asking on your data. So this is kind of the upfront general stuff that you would want to know prior to doing any of those hypothesis discovery or classifications or whatever else you would want to do with multidimensional data. Okay? So really, if you think of kind of this long tail, there's four, I guess, key steps. One is sort of this pre-processing and some sort of normalization, and then the analysis, which can be mundane to sophisticated depending on your question, and then the visualization interpretation. And I'm just going to focus today on these first two, okay? Because I think those are probably most generalizable and relevant to probably relevant to the audience in the room. Plus, this is what Holden asked me to talk about. So. Okay, so all of you, how many of you actually run some sort of fax data or have run fax data? Anyone? Okay, good. Perfect. So I thought I would start with something that maybe is familiar to you. So the idea of transforming your data is not new, right? Everyone does it in some sort or another. People may not know why they do it, but for example, does anyone recognize this equation? Who does fax data? Okay, I know someone who should know the answer. So this is actually probably one of the more common and, I guess, recommended transformations that's done. This is what's called the bioexponential. Okay? So this is the functional forms in the sort of most general form. When you actually go through, you have to solve for these parameters. And so it's a fairly complicated transformation. Okay? So you guys are maybe, while you may not know the math or how to do this, you're comfortable with the idea that you might have to transform your data. And the transformations come about because you want to help visualization. They also come about because it's very important for analysis in terms of you cannot do particular analyses unless your data is of a certain form. And so the transformations aim to put it in that form. Okay? And so some of the common ones that, the sort of simplest ones are you just scale your data in some way or add some sort of constant. Okay? And this most often comes up when you're doing some sort of background subtraction, right? So you just add a constant to subtract off the background, okay? Or move your data so that, maybe you don't want negative values, so you shift it up so the minimum value is 0 or something like that, okay? So that's the sort of simplest transformation. And then you have the logarithmic, which is a precursor to the biexponential in terms of this is trying to, if you have very disparate data in terms of the scale covers many, many orders of magnitude, transforming it using a logarithmic transformation helps squeeze the short end, the high end of the scale while expanding the short end, the low range of the scale, and actually can, in some cases, put your data into the normal range where you would want to operate it on for any parametric test. Okay? So this is something that you might not know why you're doing, but you would do it. And then commonly, if you do not do any sort of parametric testing, so a t-test, for example, is a parametric test. If you do any sort of non-parametric test, you typically do something called rank ordering, where what you care about is not necessarily the actual values of whatever your readout is, but you care about the relative positions. And are those relative positions preserved from one batch to the other, for example? Okay, so ranking things and putting them into a non-parametric form allows you to compare across batches fairly in a very easy way. Okay? All right. So as far as normalization strategies are concerned, there are many, and these mostly come. So the simplest one is do some sort of scaling, and that's averaging often by the mean, but more appropriately in many cases to scale by the median. So you're basically trying to get some average value for a batch, and you will scale your parameter to that average value. Okay? And this will allow you then to compare from one batch to the other. Okay? What's commonly done in gene expression is to do percentile matching. As you've heard of quantile normalization, this is where you rank the genes and then match the low and high, and so that you get, you're not necessarily matching on specific genes, but the ranking of the genes. Okay? And so you're trying to normalize based on the percentiles. So this was sort of the original gene expression. Now what is used often is some sort of curve fitting, where you're curve fitting between microarray chips, okay? Using either a Loess model or splines. Okay? And then what I've done and can sometimes work in, for example, the biomark data sets where you have quote-unquote housekeeping genes, or if you add a control sample to your batches when you're doing your experimental design, you can normalize by that sample or that housekeeping sample. So whether it's a housekeeping gene or a housekeeping sample. And the key thing about a housekeeping gene or sample is the underlying idea is it has to be constant or thought to be constant regardless of what happens. Okay? So then you could compare from one batch to the other. Okay? So I don't. So this sort of the nonlinear smoothing is for microarrays typically. The multiplying by a constant we typically use for Luminex and PhosphoFlow. Occasionally we'll use this for biomark data. So that's kind of the range of where these things would be applied. Okay, so I'm going to go through now in the next 10 slides or so, talk about three different areas: gene expression, which is kind of the most familiar to people, and then PhosphoFlow data, and ultimately end up at Luminex. And if you have any questions, I may, if there's time, share some preliminary stuff on CyTOF. But the basic strategy, as you'll see, for this is the same. Okay, you're going to do some sort of pre-processing step, and that pre-processing step is really to try to deal with the fact that batches differ. Okay? And the reason batches differ is there's many sources of variability, and you're trying to account for that and minimize that so that you can then compare what you really care about, which is differences between conditions in your samples. Okay? And so for gene expression, it's fairly straightforward. You do some sort of batch checking and then some sort of stabilization to the variance, and I'll talk about what that means in a second. And then the normalization strategies range from quantile normalization to these spline curve-fitting normalizations, and you basically apply them and check. And so the idea that I want to leave you all with is, these aren't necessarily solutions that you just run with and go without doing the step of what happened when I did this. So you apply some sort of variance stabilization, for example, and you see, did that do what I expected it to do? Or how did that change my data? So that's kind of the key step. So after each of these sub-steps, you want to visualize how your data was changed. Does that make sense? Have I introduced some sort of bias now that will confound my analyses later on? Okay. And just as a point on gene expression, and this is true generally, why we do some sort of variance stabilization is the fact that the mean and the standard deviation are often coupled together. As your mean increases, the standard deviation also increases. And so the variance stabilization is really an effort to control this so that you don't have differences in variance. And so your variance becomes homogeneous across your range of mean expression values. Okay, so there's a lot that's been written on this. Gene expression is probably the most developed in terms of how it deals with batches because it was realized very early on that all of your gene expression experiments were in different batches. It wasn't until recently that PhosphoFlow and Luminex came in different batches, so you didn't have to consider those effects as much. Okay? There's lots of information on gene expression, though, that you can read up on. Okay, so just as a quick example of how this works, so one of the most common techniques is clustering. And the metric to cluster on often is a coefficient of variation, which is simply the ratio of the standard deviation to the mean. So how much, given a mean expression value and the range of genes, what is that? And so is the ratio 10% of what the mean is or so forth? And so looking for the most varying genes, you can cluster those samples. So each point on this dendrogram is a different sample or a different batch in gene expression land. And you can see right off when you just look at the raw data, there's something weird about this one sample. Right? This is an outlier in that it's clustering way outside of everyone else. And even post doing normalization, it's still an outlier. Okay? So there's something funny about this sample. As it turns out in this data set, this sample is the control sample. And it turns out to be sort of a bad control in the reason that these are all 3-year-old kids, and this control is some older person. So these are all 3-year-old kids, roughly, who are sick, who are from Bangladesh. And this is some control sample that was collected from the HIMC. So probably a healthy person who's older and is not necessarily the most appropriate control. And you can see it stratifies quite dramatically. So in this situation, what you would do is you would remove the control from your pre-processing and normalization, because that is affecting the rest of your samples in a biased way that doesn't make sense for this analysis post pre-processing. Okay? All right, so and in the normalization, just to give you guys a flavor for what happens, so this is using robust splines. You get a very similar effect if you use quantile normalization. So you can see for each sample, there's 20 of them. They all have different mean expression and variance, and what you try to do is, the goal is to normalize the mean expression so it's about the same for all, across all samples. And now you can compare sample to sample or batch to batch, okay? Because now you're starting on the same footing. Whereas here, you couldn't tell if this difference here was because of the batch or because of gene expression being different between those two samples. Does that make sense? Okay. All right. So, moving right along. PhosphoFlow has been kind of an interesting challenge in that, first of all, it's immediately obvious that you, if you're going to run even a modest amount of samples, say 30 samples, where we're now, for some of the studies I'm involved in, running hundreds of samples per group, you cannot put more than maybe 13 samples on a plate, just given all the combinations of how many stimulations you want to add and how many stat or other intracellular signaling molecule you'd like to look at. Okay? So you automatically have differences in batches that you're going to have to deal with. And then the question comes up is how do you, so first you need to check for that. And then how do you deal with that? And the way that, I'll just give you the punchline right now, and of course, after each step, visualize. So just as sort of a technical point, we typically look at the 90th percentile of cells. We can talk about why that's important or the choice we made on that. It was just the choice we went with to minimize any sort of bimodality in cellular stimulation. And then we found that looking at the stimulated condition as well as the unstimulated, both of them matter. And I'll show you some evidence for that later and why you should care about the unstimulated levels as much as the stimulated levels. And then just to give away in terms of what we do, so the way we normalize the data, and this has been sort of most effective in removing batch effects while preserving signal, is to actually look at the stim versus unstimulated condition for a given sample in a particular condition, and then normalizing that by the median value of the batch. Okay? If you have control samples, you could use the control samples. They're roughly equivalent unless there's variability in the control. And then it tends to work better when you're using a group median rather than a single point control that might vary for technical reasons. Okay, does the math make sense, or do I need to describe it? Yeah.
Say again what a batch is in this case.
So a batch in this case would be a plate. Okay, so for PhosphoFlow, we typically get 13 samples on a plate, and that amounts to having 4 different cell types we're looking at: CD4, CD8, B cells, and monocytes, and then looking at roughly 7 different cytokines being the stimulants, and then looking at 3 stat readouts. Okay. Okay. All right. So, and just as a point of reference, the simplest comparison one could make is if you had a control sample in your experimental design, and you ran the PhosphoFlow, and you asked, okay, what is that control sample look like in one batch versus the other? Okay. So what would be your expected result for this? Sort of the ideal case before noise and other things interfere. Right, super. So you would expect it would be perfect correlation, and so you'd have this 45-degree plot. Now you might get some noise around that, and so this is kind of the dumbest sanity check you could do, just to make sure that something's gone well with your experiment. And so what I'm going to show you is now, I think, 9 different batches or so across. And you can immediately see, so this is the same kind of plot where you're plotting one batch versus the other. It's just when you have 9 different batches, then you get 81, or maybe it's 8 different batches, so you get a panel of 64, okay, because you're doing every possible combination. What's shown in the upper right quadrant are all the correlations for the batches. So you can see down here, in these higher batches, there's very good agreement across batches. And then you see batch 5, okay? And batch 5 has some weirdness going on where it has fairly low correlations. And these are just the same sample, okay? And I've colored these by the stat readout, so you can see that it really actually goes with maybe there was an antibody issue in terms of there was higher or lower fluorescence for a particular stat. But so this is where you would like to be, and so these are the batches you say, okay, these are great. And this is where you might say, there's some concern, and you'd have to then figure out what went wrong with the experiment. Okay? Does that make sense? And if you didn't do this, you do your analysis, and you might get some effect, or you might not. and it's going to be confounded by the fact that something's off with your experiments from the get-go. So I'll just say that batch effects matter, and you want to make sure you do them first before you do anything else, or at least look at them. Because you'll spend a lot of time doing analysis, they'll get complicated, and you want to make sure that you can trust those in the end. Okay, and then just as a point of how this works, so these were 3 different batches and this is looking at the raw data from phosphoflow samples. And if you ask, are there batch effects, and the way to ask that is just, run some sort of either ANOVA or a non-parametric test, and you can see that in more than half of in this case, there are 52 different conditions where you pair, cell type and stimulant and stat readout. More than half of them show batch differences. Okay? Whereas when we do the normalization scheme that I showed you, you get nothing. Okay? So there's now there's no batch effect. So at least when you get an effect, you know that it's due to the fact that the samples differ in some way. Okay? So I hope this is obvious, but I just want to make sure that you guys do it. Okay. And then I told you the baseline and fold change matter. So Gary is all about fold change for the most part. And I think that's truly key in that most biological processes, what you care about is the change from some condition. Okay? And I'll just point out that hidden behind this, especially with phosphostat signaling, is the fact that you're starting at some level, right, of phosphorylation. And you cannot neglect the fact that depending on where you're starting with that will influence how much fold change you get. So if you see for some reason you have lower fold change, it may be due to the fact that there's a problem with signaling, but a more obvious place to look is at the baseline and see if that's elevated. Okay, and you can see here there's a really nice coupling between the fold change response and the baseline. As the baseline goes up, the fold change goes down. Okay, that's just sort of an obvious thing, but not necessarily. I want to make sure everyone here appreciates that and just starts to think, look at the baseline in conjunction with the fold change response, okay? Or don't neglect it, I guess I'll say. All right, and then the last case study is Luminex analysis. And so again, it's the same sort of basic scheme. For Luminex, in some cases it's easier. Here, what we typically do for any sort of normalization or batch controlling is just do, again, some sort of median scaling, where it's the median value of a particular analyte for a particular batch, or you could transform it into z-scores, which has a very similar effect. Okay, and I'll just point out, typically with Luminex, the assay itself is highly reproducible, and this is kind of the same experiment as before, where we're comparing a control from the same control run over multiple plates. So in Luminex, again, you can only run so many samples on a given plate, and so when we do hundreds of samples, we have to spread them out across 7 or so batches, which would be 7 or so different 96-well plates. We know there are many sources of variability, and you want to do this up front to try to isolate that. And you can, so in this case, batch 1, 5, 6, 7, 8, 9, and 3 are all different plates, different physical plates. Okay? And then batch 3 actually has 2 technical repeats. So this is one way of sort of separating out your inter and intra batch variability, where you see there's actually very good sort of technical repeats, which you would hope. If you just pipette the same sample in 2 different wells, you would hope to get the same answer. And then, it's, in this case, highly, a very good correlation across plates. And I'll point out, just as a little bit of a plug, I don't know if you guys talked about it, but these are all done on median fluorescent intensities. The actual data for Luminex is much richer than what you typically get out, in that what's really measured are on the order of 100 beads per cytokine. And there's a new paper out just a few weeks ago by a colleague in Mark's lab, Ophir Goldberger, with statistician Johan. And they're actually starting to look at all of that data. So look for this as another way to sort of get more information out of Luminex. It becomes more complicated, but you can deal with some of the underlying problems that come with a single point measurement out of a machine. Okay? All right, so I'll leave you with this. And I've said this already a number of times, but you're going to spend a lot of time analyzing your data, especially as you're doing these high throughput assays with maybe tens to hundreds of samples, hundreds to tens of thousands to millions of different readouts. And so it's obvious yet important to do the upfront work of checking for what the batch effects are, and then trying to do some sort of normalization to minimize those. Okay? And then at each step along the way, whatever you do some sort of visualization of your data to make sure it hasn't changed in a way that is now biasing your data in a direction that confounds any future analysis. Okay? All right, that's it. Thanks.
Questions for Brian?
So a simple question. So if you have two populations of like 100 controls, 100 disease states, which for the Luminex, you take the median of controls and then normalize that median of controls? How would you do it?
Sorry, say that again? You have 100 controls and 100 disease states. And they're all spread out across different batches? I mean, you're given 100 controls and 100 disease states, and what's your question in the end?
The best way to represent the data. You normalize the median of, you take the median of the controls and then
Yeah, I mean, you could certainly, presumably those controls are spread out across multiple batches. So if it were all on one plate, it wouldn't really matter all that much, right? This is mostly for, so presumably they're spread out across multiple batches, and you would you could take the median value of each control, of the control for each batch, each plate, and you could scale that by the value of each sample. So each sample is going to. So I mean, but you check, you see if there's batch effects first of all, and then you would scale if there were, and see if that has removed it. You do the median. If you have multiple controls, yeah, the median probably would make the most sense.